We can't hear you. Could you try unmuting?
Can you see me now?
Yes. Now we
Okay, great. Sorry about this. Hi, my name is Chang. I'm from the National University of Singapore, and thank you so much for having me here today. So I think it's an exciting time to become a biologist and also a biological engineer because a lot of exciting science students are now converging into entire field of biotechnology. And one thing that particularly stands out in this era of DNA writing and DNA reading is that we can implement and execute an engineering framework that comprises design, build, test, and learn cycle into a range of biological studies and biological engineering work. So based on this, my lab is very much interested in designing the genetically encoded genetic circuits that can enable a living biological system to show new cellular functions. So very upstream, we are quite interested in redesigning and also rewriting the entire chromosomes of microbes so that we can understand better some of the key features of microbial chromosomes from bottom up. At the same time, using this approach, we can actually give a whole new functions, cellular functions to live microbes. More downstream, we also engineer microbes to produce a range of chemicals and materials in a more sustainable manner. So what we've been doing is genetically engineering the metabolism of some of the microbes so that microbes can serve as live cell factories. Another application that we're quite interested in is using microbes for human disease treatment and prevention. So in this area, actually what we're interested in is transforming some of the microbes that live in our body into live therapeutics. So as all of you know, our body is full of microbes. So every corner of our body actually contains a large number of microbes. So one of the key questions that my lab asked a couple of years ago was whether we could give new functions, biological functions and cellular functions to the microbes that reside in our body so that they could function as a potential therapeutics. So this is actually kind of a long-term vision that we laid out a couple of years ago. So my lab always aspires to develop a kind of platform technology where the microbes can be reprogrammed or engineered to manage health and disease conditions. So if we can give a microbe a new ability to detect an array of health and disease conditions, we can use microbes for disease diagnosis in our field. At the same time, if we can give microbes an ability to show some new functions that can lead to specific therapeutic efficacy, such as antimicrobial activity or even immunity enhancement, we can envision using those microbes for disease treatment. So if you zoom in for biomarkers and therapeutic molecules that can take advantage of live microbes, you can easily come to the conclusion that there are a range of molecules, including host metabolites and even external signals that microbes can naturally detect. And also there are an array of therapeutic molecules that we can engineer microbes to produce in a biological manner, so that those phenotypes, which actually show a new cellular functions, can eventually lead to very specific efficacy against diseases of interest. sound very straightforward, but one issue is our human body actually is a very dynamic environment. So if you think about the GI tract, which is about 7 to 9, 8 meters long, I mean, the intestinal fluid actually keeps moving. And also the pH fluctuates in a significant manner. And also all the interactions between the natural microbes that reside in our body and also the microbes that we actually use for disease treatment. We don't fully understand how these two different groups of microbes interact with each other. It sounds like a daunting task, but that's precisely the reason that my lab has been actually working on this for the past couple of years. And today, actually, I just want to share with you some of the work that we are working on right now, and why this type of concept might turn out to be quite useful for a next generation of therapeutics. So the complementary functions that in this particular study that we're very interested in developing in particular microbes that reside in the gut or detection of infection biomarkers, and then process this information, and then eventually showing antimicrobial activity. And also, we also wanted to give another function that allow the microbes to swim towards a specific location in the body. So this is a genetic circuit that we designed, and also we put into a gut microbe. And then what we did was confirming whether this genetic circuit would enable microbes to detect infection biomarkers coming from human pathogens, and eventually producing antimicrobial molecules, which are quite specific towards a target human pathogen, and eventually showing antimicrobial efficacy and activity. So we actually validate this efficacy in vitro. So we wanted to ensure that this activity could be sustained in animal models as well. So we want to validate in vivo prophylactic efficacy in animal models. So to test the treatment effect, we infected mice with a pathogen prior to introducing the engineered microbes. So to make a long story short, in mice, the treatment with the engineered probiotics cleared about 80% of colonized pathogens in the body. We also tested prophylactic effect, preventive effect. So we already introduced the engineered microbes and infected the mice with a human pathogen, and then we actually confirmed in vivo that this treatment with the engineered microbes actually attenuated a gut colonization of the target human pathogen by 98%. So this proof of concept study actually suggests that the microbes can be reprogrammed and engineered to detect and also inhibit infection in animal models. So in another study, we actually aim to engineer microbes to convert the metabolites we all have in our gut into drugs in the intestinal environment for cancer treatment. So for this, we designed and developed 3 new cellular functions for gut microbes. The first one was target binding to colon cancer, and the second function was the conversion of intestinal metabolites. And the last function was the production and secretion of anti-cancer compound and intracellularly produced. So in particular, we aim to convert food metabolite, so that metabolite originating from food that actually we take on a daily basis. In particular, we are quite interested in using cruciferous vegetables, including broccoli and lettuce, which actually contain a very interesting class of molecules toward glucosinolates. So interestingly, glucosinolates can serve as a stable precursor to very unstable but cancer-specific molecule called sulforaphane. So this conversion can happen by enzymatic path, but the mammals, including humans, lack the enzymatic machinery that can enable the conversion of glucosinolates into sulforaphane. So what we did was we designed a genetic circuit using the engineering framework that I briefly touched upon that comprised the design, build, and test and learn cycle. And through a couple of iteration cycles, we actually validate that this genetic circuit in this design form might be able to give gut microbes to be able to produce enzyme in the intestinal environment, and at the same time convert glucosinolate into sulforaphane. And at the same time, we actually wanted to ensure that the microbes that we hope to use can latch onto the surface of colon cancer. So we actually look for a protein that could enable microbes to bind to the surface of colon cancer. So to make a long story short, we actually found a protein originating from human pathogen, and then we expressed this particular protein on the membrane of a gut microbe. And then we actually confirmed that when this protein is expressed on the membrane of gut microbes, they can actually stay bound to the surface of colon cancer. And then, as I mentioned to you before, our hypothesis was if the microbes are given an ability to latch onto colon cancer and then convert the gut metabolite into anti-cancer compound, and then we are supposed to see anti-cancer activity against the colon cancer. That's exactly what we see on this particular slide. So, and afterwards, we wanted to also validate in vivo efficacy against colon cancer. So we used colon cancer in animal models, and then to make a long story short, so we, on a weekly basis, we introduced our microbes into the mice that actually had colon cancer. So in a span of 14 weeks, we actually confirmed that using this H&E staining assay and some other assays listed on this slide, we actually confirmed that a significant portion of colon cancer actually got eradicated by our microbes. So what we did was feeding microbes with vegetables. In this case, a broccoli pellet in water, and then engineered microbes that we designed. And then they two worked together, and they showed high degree of efficacy against colon cancer. So this work, in particular, in my view, demonstrates that engineered microbes can convert and transform normal diet that we enjoy on a daily basis into a very sustainable therapeutic regimen. And then just last study that I hope to briefly introduce to you is our more recent work on engineering gut microbes for the treatment of Clostridium difficile, which is a gut pathogen. And then actually they are very resistant against an array of antibiotics because they can form spores. And spores are very resistant to an array of antibiotics. So what what we did, and our hypothesis was that whenever antibiotics are introduced to patients, and then this balance in the gut microbiota actually breaks. And this actually leads to the increased population of the gut pathogen I just mentioned, Clostridium difficile. So our hypothesis was that the dysregulation of the microbiome-mediated metabolism in the gut could contribute to the C. diff infection. And also its restoration actually can limit the pathogenesis of C. diff infection. So this is a more biological phenomenon. The C. diff pathogen actually germinates into a more pathogenic form when the microbiome imbalance actually in the gut upon antibiotic treatment. So we aim to engineer genetic circuits in gut microbes to sense antibiotic-induced microbiome dysbiosis. But when dysbiosis happens, to increase the level of some gut metabolites. So we actually want to use them as an input signals. So in the presence of those metabolites, the entire genetic circuit is activated. And so when we hypothesized that this machinery that we designed could lead to the limiting of the germination and also eventually the growth of the human pathogen called C. diff. So this is our game plan. So we designed this genetic circuit which comprised a sensor and also amplifier and also actuator that comprises a number of enzymes and also the DNA regulating mechanisms. So we carefully designed. Of course, we have a number of different designs, but we tested, and then this design, the scheme actually, we hypothesized based on the in vitro data, might actually have best chance to show some efficacy against the C. diff pathogen. And then in vitro, we actually confirmed that this genetic circuit which I just mentioned, could give microbes an ability to detect microbiome dysbiosis and eventually the reduce and limit the growth of the C. diff pathogen. And then what we did was we validated this efficacy in animal models in vivo. So to make it almost sure that this engineered microbes actually improve the survival and clinical symptom score of mice that was our animal model infected with C. diff. So what was really exciting was the outcome that when the mice had our engineered microbes, 100% of the mice actually survived. And on the other hand, the infection actually could kill most of the mice without our microbes. So our research actually suggests that microbes engineered to modulate the intestinal metabolism actually can really inhibit significantly the C. diff infection in animal models. So let me just give a few slides, and then let me to my concluding slide. So this is just possibility. I mean, there's no FDA-approved live biotherapeutics yet. Even though a couple of clinical trials are underway, mostly in the States. But we're actually making some platform technologies right now so that where we can actually give an array of new functions to the microbes that reside in our body. So we're studying gut microbes. We're also studying skin microbes and the microbes that live in different parts of our body. Our question was, can we give new function to those microbes? And of course, we actually have to study how they really behave in a more biological manner. So that's another aspect of our study. So we study and we come up with a hypothesis, and then to test the hypothesis, we actually design and develop new biological functions. And we envision actually using this cycle and this framework to treating an array of human disease conditions, including cancer and metabolic disorders. With that, I'd like to thank my funding sources. Let me just, my funding sources and also my group, and thank you for your attention.
Thank you, Professor Chang. Any questions from the audience? Let me ask a question. Can you hear me well?
I can understand if you ask, but yes.
Okay.
I'll try.
I'll try to speak up more. So,
But now I can barely hear. Sorry.
no? Probably I'll just speak in the room, and I may type that question in the chatting window. Okay? So you showed that you mentioned that your microbes, some of them are on preclinical or clinical trials in USA. That how would you envision that we can swap?
Right, yes, that's an excellent
microbes?
question. So, as I mentioned to you, this is a new modality. I mean, this is a kind of new intervention strategy. But FDA rolled out the regulatory path and also guidelines in 2018. So since then, a couple of clinical trials got underway, as I mentioned before, mostly in the States. So actually involve this type of microbes. So the microbes that have been engineered and then given specific therapeutic functions. So as I mentioned before, a couple of clinical trials underway, and then some of them actually have passed maybe phase 2 in a very successful manner. So they're actually pushing towards a regulatory approval in the coming years. So that's where the entire field is right now. But again, there's no FDA-approved live therapeutics that actually comprise engineered microbes yet in the market. So there was another question about the delivery mechanism. So if we had to target gut diseases, so I mean, as I mentioned to you before, in our animal models, there's a water delivery. So we use a powder, so lyophilized microbes, the freeze-dried microbes, and then we just mix them in water, and then eventually the microbes in this form or the route actually can reach the intestinal tract. But eventually, for translation, of course, we actually have to use a slightly different strategy. Yes, the microbial drinks, yes, like a yogurt. So we can actually use this type of engineered microbes for the same purpose.
Okay, thank you. Any other questions from the audience? If not, let's thank Professor Chang again.
Thank you.